impressive, but at the same time, sporadic history of life on Earth as we can find. Now, to find a fossil, you really need to have something that tends to be aquatic because it has to get killed or die and then get covered in sediment typically relatively quickly. And then over time, that sediment builds up over top of it, and you can get where the sediment becomes sedimentary rock, or you can get where the substance that's in there that's buried becomes mineralized. If it's bones or teeth, it can be preserved until it's found. And so there's certain places that are more likely for this to happen. Aquatic is one of them, tar pits, desert areas, or glaciation as long as it sticks around long enough. Obviously, if the glaciation's there and then it melts, you're no longer frozen. And so at that point, you can start to decay, but it can help preserve stuff at least for a longer period of time. There's also insects that get stuck in amber, which will not help you make dinosaurs, but there's a variety of different things like that as well, footprints, all kinds of stuff. So other things that can help you get a fossil would be if something has a long existence, and what I mean by that is throughout the history of the Earth, not like you live for 1,000 years. Same thing with abundant and widespread. It just means you have more opportunities for fossilization to occur. More opportunities that someone dies while in just the right conditions, where they fall into a lake if they're normally terrestrial, or they fall into those tar pits, or it's dry out for that period so they get submerged by sand or debris before they can ultimately decay. You also have hard parts is probably the best option. And so if you have harder parts, these tend to persevere longer, so it gives it more time to stick around and get covered and end up in the right conditions to allow fossilization to occur. Now, even on the rare chance that you actually get fossilization, and it's rare even if you've got all these things, you still also have to find the fossil. And so over time, you can get where rocks are exposed due to erosion, and fossils can be lost because now they're exposed to the elements, and so they can wear down. And so it's not only kind of a big deal that something fossilizes, it's also a big deal that we can find the fossil. Now, these are just some examples. This is an ammonite, which is like a shelled cephalopod, like squid octopus type thing. So these guys had a hard shell, and they were very plentiful for a long time. And so these are an ideal example. These are trilobites, which are, I don't want to say an ancient insect, but they were somewhat similar to like an insect or a crustacean. And once again, these had a hard exoskeleton. They were very plentiful for a very long time. You're talking probably a couple 100 million years. And so we see lots of these fossils because they met all those criteria to be likely to be fossilized. Now, we also need to put these in order, so we have to have ways of dating them. I'm only going to go over a couple. So we've got relative dating, which works by the law of superposition, which says that these strata that we see, these kind of lines, were different sediments put down at different times, and the youngest will be at the top, and the oldest will be at the bottom, because that was put down first and the other stuff's been put over top. Now, occasionally this will break if you have like movement of the crust areas, so you can have where there's upheavals because of earthquakes or mountain building, so you can't always do this. You've got to make sure you pay attention to see if things have shifted, if something's been shoved upward. But in general, this is what you'll see. And so if I find something here, I can assume that it is younger than whatever I find here. And if I find something down below that, I can assume that's the oldest. Now notice this doesn't give me an absolute age, it just says it's younger than so-and-so or older than so-and-so. Absolute ages work by trying to give us a more accurate age using radiometric decay of radioactive isotopes. And so what happens here is isotopes that break down radioactively do so at a set rate, and that's called a half-life. And so after one half-life, half of the stuff will remain. And so you can see here we have half of our original radioactive sodium, and the rest of it's converted to the daughter isotope, which in this case is neon. And so we can see this 50-50 ratio, which lets us know it's been one half-life. In the case of the half-life here, it looks like the half-life's 2.6 years. We have lots of different radioactive isotopes that we can use. So some of them, like radioactive carbon, is going to be in the neighborhood of about 5,730 years, I believe. So that one's good for stuff that's about 50, maybe 60,000 years old. And then we've got stuff all the way through uranium that has a half-life of 4-point-something billion years, things like 4.2 or something. So really long half-lives, which are good if we're trying to find out like the oldest rock. which is about 4 billion years. And so we can look at that. And we typically, when we're doing this, we'll find igneous rock that's been made at that time that ends up in the same strata. And so we'll find several different pieces of this igneous rock and test that, and then use essentially relative dating to say, then these other things, these other fossils must be about that same age. So keep in mind, you can't directly absolute date fossils typically. You're going to be absolute dating the stuff that's surrounding those fossils in the same layer. So that's going to be one of our ways. And you can notice here too that as you go on, half of 50 is 25. So after another half-life, 5.2 years instead of 2.6, it's 25. And then from there, you're going to see 12.5, which is 1/8. And then after that would be 1/16, 1/32, etc. At some point, you have so little of the original radioactive isotope left that we just can't use it. So we'd have to move on to an older, or I should say longer half-life radioactive isotope, which would give us more accurate data. Just like you don't want to use something with a huge radioactive isotope for something that's a couple thousand years old. All you're going to see is it really hasn't decayed much. And the last one is useful in certain circumstances where we can tell that the Earth's magnetic field has flipped and traveled several times, well, often over the course of history. And so especially if it's more recent, we can use which way rocks that are formed that kind of imprint that magnetic field. We can look at those magnetic fields to kind of figure out exactly what time it was, because we can try and merge these other types of aging with the records we have of the magnetic field and what it was at various stages throughout history. So I don't know personally if this would be very useful stuff really long ago, but this is another one that would be useful at least in recent terms. There's also tree cores that you can look at. There's also digging through and coring ice stuff in the glaciers. There's lots of other ways that you can go about doing this, but these will be some of the big ones that we'll talk about, especially relative and absolute dating. Now, mass extinctions are big events that typically wipe out about half or more of the aquatic species that exist, the marine species. It can also really mess up land species too. In the past, there have been 5 big mass extinctions, and ultimately there's some thought that we might be heading into a 6th one now. But I just want to make sure you realize these can be huge times that oftentimes can be climate change. Some people theorize there's hydrogen sulfide gas that's released that wipes out lots of stuff. You can obviously have like a asteroid or comet or something big that smashes in, but something big happened around these times that really wiped out a lot of life. And so as you go back, you can start towards the beginning here. You've got the earlier times. And so we've had several mass extinctions. You had they're usually named for what came before it. So we had the Ordovician mass extinction here, the Devonian mass extinction. The Permian was probably the worst. This one wiped out over 90% of marine species and probably over half of all the land species that were there. This one, I believe, is the one that killed the trilobites. It messed up some stuff that was very, very common up to that point. It also likely set mammals back because mammals were just starting to kind of get their groove on here, and they were competing with the reptiles, but the mammals were disproportionately affected. We almost went completely extinct. 1 or 2 species snuck through, and so that was that. You then had a relatively small, compared to that, Triassic mass extinction, and then the last one was the Cretaceous mass extinction that wiped out the dinosaurs. So those are just some of the big mass extinctions. And then after that, you actually have a lot of options, because when all these guys disappear, you see adaptive radiations of the guys who are left because there's so many niches available. Because the guys that tend to die the most are the big guys. And so now as things stabilize, the small guys can get big. And so they can take over a lot of those things that previously they did not have access to. Now, the first eukaryotes, when we make this transition, is just going to be our shift here where we started to see pieces of membrane kind of get pulled in to cells, and those became membrane-bound organelles like ER, the Golgi apparatus. They could be things like nuclear membranes, so it let us compartmentalize. But we also have something called serial endosymbiosis, where it looks as though some aerobic prokaryotes got ingested by bacteria, and they basically live symbiotically with it inside, and eventually horizontal gene transmission moves some genes to the nucleus to give up control, and so we got a mitochondria, an organelle instead of a cell. And then later, some cyanobacteria did the same thing, multiple times likely. That's why we have different types of algae. And so those gave us photosynthesis. Those gave us eukaryotic cells that would be more like a plant cell or an algae cell. And so this event did happen multiple times. You can also get other plastids like this, where other things migrated in through that way. But there's a difference. Organelles that are just a membrane, they're infoldings. But if you have your own DNA, your own ribosomes, those guys appear to be this endosymbiosis. Now, the evidence for this is that a lot of these organelles have two membranes, and the inner membrane is very much like a prokaryotic membrane. So it's as though it got ingested by a cell, so the outer membrane is like that of the cell itself, but the inner membrane is like a prokaryote because it's like a prokaryote that got eaten. The organelles themselves go through a division process that's very similar to like binary fission that you see in prokaryotes. They have their own DNA and their own ribosomes, and the ribosomes are like the ribosomes of that of a prokaryote. They're not as similar to those of eukaryotes. So all of the pieces that are in these organelles, like mitochondria and plastids, like the chloroplast, they basically resemble little prokaryotic cells to this day in large part. So there's a lot of evidence that supports this idea of endosymbiosis, even if we can't go back and see it. And plate tectonics, as we go through a quick crash course here of various things, plate tectonics, this movement of the continents, has had a big effect on evolution, because as these things move, it leads to environmental change, but it also influences migration and how things can move. So for instance, you had a while back that marsupials developed in what was likely South America, and then from there they migrated through Antarctica, which at the time was a decent place, it wasn't horrific, and migrated into places like Australia. Australia then broke away, Antarctica became a frigid, terrible zone, and everything died. And eventually South America connected with North America, and so many of the marsupials couldn't hang on when facing the competition of things like placental mammals that could move down through there. So a lot of the history of things, a lot of the biogeography, a lot of the reasons for things can be explained through plate tectonics as to why these things happened and why animals had to perpetually cope with things. They didn't have just 300 million years where they could sit back and relax because things stayed the same. There was a lot of change that then drove this selection process that allowed for evolution. And the last chunk of this chapter, which is a fairly large one as you can see, is how do we get some of these new structures? And so we're just going to discuss some of the ways we can get changes in an organism to give us some of these new possibilities. So one of the first ideas is exaptation, where if you want to get something new, use something old that you don't necessarily need. So you can see there's many different ways of using webbing that we've built to get wings. These are exaptations. The webbing was not useful for flight as it was. That wasn't its original purpose. But the fact that you can have webbing, which is more likely back in the day, if you will, to be used for swimming, the fact that we have that kind of within our realm of possibilities of genetic malfunctions or genetic expression allowed for us to kind of use this to eventually fall with grace and then glide and then fly. And we see different versions of this in the pterosaurs, the bats, and the birds. And so there's other cases of this that you see with insects where they might have adapted things like gills for these purposes. There's a variety of things or penguins where they have fins that are essentially were wings that they've now adapted for swimming. And so exaptation is just this process of taking an old structure and kind of repurposing it for a new structure, not trying to build something from scratch. Now, other ideas we have are we can really mess around with this developmental process. So they call this Evo-Devo. So Evo-Devo is looking at how things can be manipulated during this development process, kind of like when we talked about embryology, to cause pretty significant changes. So heterochrony is kind of changing the rate and the timing of developmental events like growth. And so you can see heterochrony here when you're a baby, they're just like, let's do all head. Let's just aim a lot of our development to making a gigantic noggin. So you've got big old noggin, not a very large body. But then as time goes on, we can see we change that developmental pattern, and so now our noggin is not particularly huge relative to our body because we're growing our body at a much faster rate than we'd be growing our head. And so if we manipulate this, though, you could fairly easily get, a group of organisms, you could say, that have like a gigantic noggin and also have a gigantic body if you kind of made everything grow together at the same rate. You could have where a person, like achondroplastic dwarfs, where they have ultimately a normal torso and head, relatively speaking, but short limbs, because you just change the rate and the timing of things so you seal off those bones and you leave them smaller. And so we can get what appears to be very significant changes just by messing with when and how gung-ho we are about these developmental events early on and throughout even maturity, up to maturity, these developmental events. Paedomorphosis is kind of tied to this, and this is where you can retain juvenile characteristics. So you could have something that perhaps as like a tadpole for frogs as a larval form, and they could retain some of those larval characteristics even into adulthood. So that could explain how they could have a tail. Or salamanders oftentimes have gills when they're younger, and some adult salamanders retain those gills. So that's paedomorphosis. They keep some of those juvenile traits into adulthood. And so really all you have to do is make sure that you reach sexual maturity prior to getting rid of those traits. And if you do so, you're essentially an adult with those traits. And many of them will stay with those organisms permanently, allowing, for instance, the salamander to stay fully aquatic throughout its whole life and not have to worry about breathing through lungs. And then homeotic genes are one of the last ones here, and these are oftentimes called the architect genes, because these control what you put where and how many. So they don't say what type of leg you're going to have, but they can say, let's put 2 legs here, or let's use 6 legs and put them here, here, and here. And so if you mess with these homeotic genes, you can fairly easily get like repeats or stutters. So it makes something like a millipede be a lot easier because you're not building a new set of legs, one by one by one. You can instead just have this gene that says, all right, we've got all these genes that code for a segment of the body. What I want to do is let's do 100 of those segments instead. And so you end up with this 100-segmented organism, each with 2 legs and all the organs it needs, instead of just having one that was maybe 2 or 3. So these homeotic genes are very cool, and they allow for a lot of major change with only minor mutations. It's also cool because some of these appear to even be in common, where they can take genes that code for like eyes and move them around between fruit flies and mice, and they still function. They still say put 2 eyes here, and they don't code for what type of eyes. So if you're in a mouse, it says put 2 eyes here, and it puts mouse eyes there because that's what the genes of the mouse say. If you're in a fruit fly, it puts fruit fly eyes there, compound eyes, because that's what they say. And so as long as the gene itself that says where to put them is the same, you don't have to worry about the other stuff because those are separate genes. The mouse still codes for a mouse eye. These homeotic genes just say where and how many. But if we screw that up, I suppose we could have like an 8-eyed mouse. I'll look forward to that. And then the very last thing we have here, this is the last slide I promise, the major lineages of life. And so when we look at life, we're going to break this up into 3 major groups. So the 3 major groups are going to be bacteria, or sometimes called eubacteria. This is the first domain, and it'll be prokaryotic. You have the second domain, which will be archaea or archibacteria, as they might say. And then the third domain is going to be eukarya, or in this case as eukaryota, whatever, we'll go eukarya. I don't normally see it that way. I'm going to cross that out so it doesn't bug me. And so within eukarya, you'll see we have 4 kingdoms that are widely accepted, although pretty much everybody says Protista needs to be broken up into more. Right now there's just some debate over how many kingdoms to break Protista up into. We'll talk about that more chapter 26 when we get in classification. But there's the whole domain idea here is we only use 3 domains to try to emphasize that the eukaryotes are all fairly similar. The prokaryotes, if you look at them molecularly, I know they look similar because they're like one cell, they're very different. And so that's why they've made 2 domains where each domain just contains 1 kingdom. And the kingdom is essentially bacteria. The kingdom is archaea. It's the same thing. And so by doing that, they try to show the overall differences that we see. And you can also try to see some of these connections, where overall we appear to be most similar to archaeans, although there were likely perhaps some bacteria that moved in that became like organelles. But if you want to see who we're most closely related to of the prokaryotes, it's like these guys, the archaeans. So we're going to discuss those more during the classification chapter. I'll go more in depth with this. And we're also going to pick up a little bit more in chapter 27 when we talk about prokaryotes. I'll bring up some of the differences between eukaryotes and prokaryotes. Hope you guys could tolerate this. Sorry it's long. Bye.